Está iniciando ali. Tá ótimo. Bem, bom dia, boa tarde, eu não sei muito bem. Boa tarde. Eu me chamo Jefferson, com Eco Feedbacks. Venho trabalhando muito com guppies em Trinidad. Agora a gente está desenvolvendo um projeto em diferentes ilhas, tentando entender como organização secando alguns peixes. E hoje ele vai falar sobre... Are you talking about Trinidadian stuff? Yes. So, hoje ele vai falar sobre vários dos projetos que ele teve em Trinidad. E welcome, Andrés. Thank you so much for being here. Thank you all for being here as well.
Obrigado. Thank you. Thank you very much for being here. So today, I want to talk about some things that I've been thinking about lately regarding, well, okay, I'll start by telling you that my main interest in ecology and evolution is eco-evolutionary feedbacks. I am, by training, an evolutionary biologist that is venturing himself into ecosystem science because of my interest in eco-evolutionary theory. The whole idea is, as an evolutionary biologist, traditionally we see things like individual adaptive traits and we think, how are they a response to selective pressures created by the environment? But in reality, if individual traits change because of evolution, they are going to change their ecosystem function and what they do to their environment. So, in reality, it's a two-way process where environments select for different phenotypes. As these phenotypes change, those phenotypes may interact differently with the environment. You may have a different environment that exerts now different selective pressures, and the feedback goes on until it finds equilibrium or alternative states or whatever. And if you look at these things theoretically, they can be a lot of fun. In particular, today I'm going to talk about one of my latest interests, which is nutrient recycling. And nutrient recycling can happen. So, in terms of the effect of individuals in the ecosystem, I'm interested in nutrient recycling and how those traits that give nutrient recycling evolve. Okay. Now, there's two ways nutrients can be recycled. One, you can die and be decomposed. Not much evolution there. Or you can do this thing that we call homeostasis. Okay. Homeostasis, in particular stoichiometric or elemental homeostasis, is the idea that organisms have a given chemical composition, right? And the whole point of life is that you keep that chemical composition despite the changes in the environment, right? I mean, many people consider that a very definitional aspect of life, the ability to keep themselves away from entropy, away from just dissolving and being the same composition as their environment, right? So what happens, how does this done? It's done by either acquiring resources that have the chemical composition that you need for your body, or assimilating differently the resources that you need the most. And also, on the other end, you can digest or defecate, like we call it in more regular terms. So take out whatever hasn't been assimilated, or you can excrete whatever has not been metabolized is excreted. It's when you do this latter part that things get recycled, right? So the funny thing is, the interesting thing about understanding homeostasis is that inevitably, life has to alter the chemical composition of its environment to keep itself alive. For this fish to be alive, for this fish to keep its C, N, or P the way it wants it, regardless of the C, N, and P in the environment, it needs to do things to alter the environment by eating selectively, by excreting selectively, etc., etc. So I found this to be a very interesting fact. I know it's very obvious for many of us, but I found this to be very interesting because it ties very nicely to the whole idea of eco-evolutionary theory. The idea that if you involve different traits, say different stoichiometries, different acquisitions, different ingestions, different excretions, you are changing your environment differently. Okay? And that's what interests me lately. Now, let's keep talking. Let's talk a little bit more about homeostasis. Homeostasis is broadly defined as this ability to remain a certain constant, more or less constant, or stable would be better term, a stable inner environment in the face of changes of the outside environment. That could be with temperature. We keep our inner temperature more or less around 36 degrees, regardless of the temperature outside. And other things can be, like in other cases, you can have chemical homeostasis, which is what we've been talking about. If you plot here, for example, talking about chemical homeostasis or about elemental homeostasis, if you plot here resource stoichiometry, so the chemical composition of what you're eating, against consumer stoichiometry, your stoichiometry, your chemical composition, you could draw a straight line, a one-to-one line, in which you are what you eat. You exactly are the same thing as what you eat. Most of the time, that means you're dead, right? Normally, being alive requires you being, is the idea that you are different from your environment, right? You have a certain integrity in spite of your environment. So that's no homeostasis. Then you have what we call strict homeostasis, which is the idea that no matter what you eat, the way you assimilate, the way you select your food, the way you excrete, your chemical composition is going to be the same. The analogy with temperature would be homeotherm, right? No matter what temperature it is, the moment you are the temperature of the outside, means you're dead, right? But not everything is so radical one way or the other. We know, for example, with temperature, the reptiles don't keep exactly the same temperature all the time, right? But they're also not exactly the same temperature as the environment, right? So there's some middle term. This is what we will call flexible homeostasis. It's the idea that if your resource stoichiometry or your environment changes, you change, but within a certain range and within certain bounds that are, amenable to living, right? So, as you can imagine, flexible and strict homeostasis could have very different impacts on the ecosystem, right? Here you have individuals that are, for example, imagine that you have few resources, little phosphorus, right? And individuals are trying to keep a high level of phosphorus, organisms. They eat all the phosphorus they find. They try to excrete the least possible to hold on to it. So they become like, they sequester phosphorus, essentially. They become, the word is escaping me now, sinks of phosphorus, right? But if it's more flexible homeostasis, maybe the recycling is more dynamic. Okay, so this is why I'm interested in this evolution of this organismal aspect that has an ecosystem consequence. You can then redefine the eco-evolutionary feedback theory in terms of stoichiometry by saying that the biogeochemistry of an environment is what determines, through selective pressures, the stoichiometry of organisms, which in turn, through homeostatic processes, affect back their environment. Okay? Oh, you can interrupt me with comments, questions, whatever. I can say it. I'm okay with the dynamic talk. Okay, so if we're going to start eco-evolutionary stoichiometry, if I want to build myself a research program on eco-evolutionary stoichiometry, what kind of things do I need to establish at the beginning? First is, what are the selection pressures behind stoichiometric traits? Right? What is causing individuals, or what is causing populations to be more homeostatic or less homeostatic? What is the advantage of one or the other versus different resource environments? Of course, if we're talking about evolution, these traits, the stoichiometric traits, the ability to do certain homeostatic processes, or even the chemical composition, it says, needs to be variable and heritable. If not, there's no evolution. And another fun, important thing is, can it evolve rapidly? Because these feedbacks become a lot more dynamic and interesting to study when evolution happens rapidly, and they become more relevant because if evolution happens at ecological timescales, the feedback is stronger. Okay, so can they evolve rapidly? So today I'm going to give you some examples of projects where we've tried to sort of like hint at these three things. And of course, this is very early on in the project, so they're not going to be final results, and they're not going to be great answers to the questions, I'm afraid. As some of you may have already known from what Jefferson said, or no, if you listened, I work on Trinidadian guppies, which are a little fish that is native to the island of Trinidad and Tobago, and on some parts of the Orinoco Basin in Venezuela. but it has been introduced for very poor mosquito control all over the world, including Brazil and Rio, which is why I'm here. Because we started, well, Jefferson is starting to look at them in their invasive range. But this work is all going to be Trinidad. Let me tell you a little bit about Trinidadian guppy for those who are not familiar with the system. Evolutionary biologists like working with guppies because they have a series of traits that make them interesting and amenable to work. They have two distinct, in Trinidad, they have two distinct phenotypes or ecotypes, you could say, associated with predation. You have high predation phenotypes and low predation phenotypes. High predation phenotypes, well, I'll tell you what they are a little bit later, but just think about two different phenotypes adapted to either predation or no predation. The differences are in life history and other traits that we have studied. So, for example, you can imagine high predation traits are those which reproduce fast. They put out a lot of babies, small babies. Okay, while low predation, they reproduce slower, they mature slower, they produce fewer, bigger babies. Okay, there's other traits like coloration that differ, like physiological traits that differ, behavioral traits that differ. But the nice thing is that they have these differences have occurred multiple times independently. If you look at here a map of Trinidad, you see all these streams. All these streams are divided by a big waterfall. It's like a little rift here. And there's a big waterfall in every stream. Above, there's no predators. Below, there's predators. Every single low predation site has been colonized by the high predation guppies from below the waterfall. And every single time they've gone up a waterfall, they have evolved the typical trait of a low predation guppy. So we can find this a repeatable event, evolutionary event. Okay, so that's why evolutionary biologists like it. But what's most fun of all, although we stopped doing it, is that they can evolve rapidly after you translocate guppies from a high predation environment to a low predation environment that has no guppies. They will evolve all those traits that I've talked about, those lower life histories, the coloration, everything, really just a couple of years. Like coloration can be a couple of years, life history about 10 years. Like it's really rapidly. So we can actually study evolution live as it is happening. This is why evolutionary biologists love working with this. So if any of those traits have ecological relevance or ecosystem significance, we are hoping to see how they are evolving in real time as we translocate the guppies. Okay, a little summary of what things are different in high and low predation environments. We talked about fast versus slow life histories. But the other thing that you need to think about is that what happens when there's no predation? There's more guppies. So then you have higher densities. If you have more densities, what happens? There's less resources per capita, right? So the adaptation to low predation is not so much a low predation adaptation as a low resource adaptation, right? Resource limitation adaptation. That's what we're starting to get interesting in terms of ecosystem things. So the counterpart of predation is often competition, right? You have less predation means you have more competition. If there's more deaths, you compete less. If there's less deaths, you compete more. And as I said, when I was talking about the different streams, how they go colonize from downstream to upstream, the high predation phenotype is always the ancestral to the low predation phenotype. And genetic data has confirmed that. All right, so let's think about what kind of traits should we look at guppies in terms of like their effect on resources, right? And by resources, eventually it would be the chemistry of resources, right? This is work done by your beloved Eugenia Zan. Was interested in seeing what was the different. Well, she was interested in many things, but one was the difference in the diet between high and low predation guppies, right? And what she found was if you went to a stream and cut open a bunch of guppies and looked at their gut contents, in high predation, they have a lot of macroinvertebrates in their guts. Well, in low predation, they have a lot more detritus and algae in their guts. So in low predation environments where food is more scarce, they eat more crap. Okay, so that was actually a pretty interesting result. One of the things that actually was really interesting that she found too is that the more crap they ate, so the more invertebrates they ate, so the better quality food they ate, the more invertebrates in their gut, the smaller their gut was. We all know from basic physiology, digestive physiology, that like if you eat mostly detritus and plants or primary producers, you need a longer gut to assimilate all that stuff, and in the middle of all that carbon. So that was a nice, interesting result that showed like some sort of like anatomical difference to these that represented this ecological function difference. So that has to do with acquisition and assimilation, right? The part that goes into the fish. Now, how about the stuff that goes out of the fish? This is excretion values. This is predicted values from the models we fit. We have here mass. Like every physiological trait, excretion allometrically scales with size. So the bigger the size, the more they excrete. But you can see that for nitrogen here and phosphorus here, high predation guppies always excrete at higher rates than low predation guppies. Okay, so think about this again in terms of what environments they're in. Low predation guppies have less resources, less nutritious food, so they hold on to their nutrients more. Okay, so that's potentially an ecological consequence there. As a result of all this, another thing that if we look at the body stoichiometry, this is the level of imbalance. So what's the difference between their stoichiometry and the food stoichiometry? More imbalance means you need to eat more food to get the same amount of nutrients you have, because your food is low in nutrients while you're high in nutrients. Okay, while if you're more balanced with the food, it's lower in balance. And these are pairs of streams, high and low predation, white being high predation, black being low predation, and low predation is consistently more imbalanced, which goes along with what we have talked about. This is for phosphorus, actually. We talked about that they are in a more limiting environment, resource limiting environment. Okay, but all this is field data. If we want to talk about evolution, we need to know, are those differences just purely environmental, plastic, or are they genetic? And if so, like what determines that difference, right? So here we have two of our great students, Amina Mohamed, graduate student, Limisha Gautam, undergraduate student, and my collaborator in just about every project I do, also my wife, Swan Gordon, working on this project. And we were thinking, okay, we expected that if you have, there's this theory out there that says, if you have limited resources, you should be more stoichiometric. More stoichiometrically homeostatic. You should be more homeostatic because you have to have a body that's independent of the crap that's outside. You need to maintain yourself at a better situation than the outside environment. If you can pick any food you want to be whatever you want to be, you don't need homeostasis, right? You just pick the right food and it's good. You don't need to for example, if you have too little resources, excrete less, because it's more limiting. So we expected low predation guppies to be more homeostatic if this is resource stoichiometry and this is consumer stoichiometry, like the graph I showed you before, that if we gave different resource stoichiometry to guppies, the low predation guppies, which are used to, struggling with resources, should be better at maintaining an inner environment than high predation guppies, we should just go more with the flow, whatever they are, what they eat, and let them know. Okay. Everything, does this sort of hypothesis make sense to people? Okay. So what Amina and the rest of us did was, Amina designed artificial food to look exactly the same, the two types of food, high and low quality food, but it was mimicking the stoichiometry of either triazine algae or juice invertebrates. Okay, and she tried on food on these two types of foods, high predation guppies and low predation guppies. She did this with third generation guppies from the field, meaning that once you have raised them in a common garden, you take away the environmental influences and you're just looking at genetic differences. And she also did a split brood design where she would take siblings and would put half of the siblings to the high quality food treatment, half of the siblings to the low quality treatment. So we were assessing here genetic differences and whatever's going on. And these are our results, or preliminary results from one of the populations. Phosphorus, nitrogen. What do you see here? In phosphorus, you see that there is a clear increase in the amount of phosphorus in the fish when there's a higher amount of phosphorus in the diet. So flexibility, flexible homeostasis. Okay? While this is a non-significant decrease, well, there's essentially no change in the amount of phosphorus in the body of the low predation guppy when you change the chemical composition of the food. Now we look at nitrogen, and we actually see the opposite. High predation guppies maintain a constancy over changes in nitrogen content in the food, while low predation guppies change. Now we said that low predation guppies were more resource limited, right? So resources, of course, people who would do persistent science here would say, oh, resource is a very vague statement, right? Are you talking about nitrogen, or are you talking about phosphorus, which is the limiting nutrient, right? So we actually went out to look at master species that has never been published and look at the limitation, the phosphorus and nitrogen limitation in this stream in high and low predation environments, and saw that these streams are phosphorus and not nitrogen limited. When phosphorus is the limiting environment, our hypothesis, our limiting nutrient, our hypothesis holds. The more stringent, the ones who live in the more stringent conditions are more homeostatic than the ones who live at less stringent conditions. Nitrogen is more limiting downstream in the high predation sites, so you find the opposite pattern. So we are quite excited about this, but this is one stream, one population, high predation and low predation. We're going to repeat more streams, and we're going to be also repeating it in streams where things have been, guppies have been introduced to see how if this has evolved rapidly, these patterns have evolved rapidly. But I told you this is mostly preliminary results. We're excited about it, but you know what happens when you repeat things, so I don't want to jinx it. Other things that Amina looked at is the excretion rates of these fish. And what she found is that, well, in both cases, the more the food quality, the more they're excreted, which makes sense, the more excess they have of that nutrient. But the nice thing we found is there was a genetic difference between high and low predation. So the difference we had found in the field of excretion rates, we can actually replicate in this lab, second generation full sip design, half sip design, which means that the differences in excretion rates are genetic. That is a trait that could evolve. So that's something that we're also very excited about. Now, can things evolve rapidly? Well, I told you we're going to redo this with populations that have been introduced, but we haven't done it yet. What we have done is there was this series of wonderful undergrads that did an experiment, Shauna, Alex, and Nana. And they did this experiment with Amina and, I don't know if you know this guy. And again with Dr. Swan Gordon. And the idea was those results we find in their guts, where they seem to eat more like juicy invertebrates in high predation and more like detritus and algae in low predation. Is that reflected in a trait that evolves? So, for example, like food preference. Okay, do they prefer that food, or is it just they eat what's there? So this goes into the acquisition part of the equation, right? So one of the things in my life that has given me the most white hairs is this long-term experiment. Gray hairs, sorry, I should say. It's this long-term experiment that we started in 2007, where we took high predation guppies and we put them into low predation sites. And this is how to explain this. Yeah, so we put them into low predation sites and we waited for them to adapt to the low predation. So we have some before introduction data, some after introduction data of all sorts of ecosystem parameters. We also left a part upstream without any guppies to keep as a control, to keep any annual changes in El Niños and La Niñas and whatever like that we have as a control to compare whatever's happening here. So it's a backy design for those who are familiar, before after control intervention. Then we let evolution happen. And we're measuring all this. There's people looking at ecosystem stuff, evolutionary stuff. I mean, there's dozens and dozens of people working on this experiment. And we did that in 4 streams. Okay, so it's replicated 4 times. Among other things that are done is every single month, students go there and take every single guppy out of the stream, mark them individually, genotype them, and release them again until the next month, when they catch all the new ones and mark them again. And this has been happening since 2008 without stop, except COVID. Where let's say it's a year and a half of missing data. Right? So great power to look at evolutionary, like what are selection pressures, because you can measure fertility, mortality, traits, etc., etc. But in this case, we made it simple, and all we did is took individuals from the ancestral population, so they look like the ones we put in, and individuals from the derived population of those streams, about, I think it was 12, 14 years after the introduction. Raised the two, the ancestor and derived, in a common garden experiment for 2 generations to make sure we remove any environmental effects. And then we measured their behavior, okay, their feeding behavior. We gave them an acclimation time. We presented simultaneously 2 foods that look exactly the same but have different stoichiometries. The higher quality with more nitrogen and phosphorus, and the lower quality with less nitrogen and more carbon. Okay? And for 5 minutes, we let them do their thing and then counted how much time they spend in each of the foods. Okay? And this is what we found. Let me walk you through this graph. Here we have low quality food and high quality food. Okay? Here we have high predation guppies and low predation guppies. And this is the time spent on each of the foods. Okay? So what happens to high predation guppies? No matter, so this is time feeding on high quality food. This is not low quality and high quality. This is the first food approached. Because we need to control for the first food approach, because sometimes they just continue with whatever they started. So you need to correct for that. And what happens here is that no matter what food they approach first, high predation guppies choose a given level or a given preference for high quality food. Okay, they like their high quality food no matter which one they found first. Low predation guppies, if they started with low quality food, they stayed on low quality food. If the first thing they found was high quality food, okay, they stayed with high quality food. Essentially, what this is telling us is that high predation guppies have a preference, regardless of what they find first. Low predation guppies eat whatever they find first. Okay? And this is a genetic difference. So essentially what this is telling us is that the introduced fish have lost their pickiness, their food preference. Okay? They eat anything, whatever is available. And that's a genetic difference. So this is rapid evolution of a food preference trait. Okay? Okay, so I've been talking about all these things. What do they all mean? Okay, I asked the question initially, what are the selection pressures behind stoichiometric traits? That was a key question to study eco-evo feedbacks in stoichiometric context. Perhaps it's resource availability or resource limitation, like Amina's experiment seems to suggest. Are stoichiometric traits variable and heritable? We know that excretion and homeostasis and even food preference is. So that's good. That means you can study their evolution. Can they evolve rapidly? We know that at least food preference can. And we're going to look into the other traits. So, and with this is all I wanted to tell you. I didn't want to tell you too much because I always appreciate feedback. So, especially with early projects like this one. So, I'm hoping we have some time for that. This is a lot of people I'm talking on behalf of. On the evolutionary side, we have all these people here. On the ecosystem side, I put you on the evolutionary side, Jefferson. I mean, I hope you're okay with that. On the ecosystem side, we have a lot of people, and of course, there's a lot of feedback between us. And of course, to my favorite field assistants. Thank you. If you have any questions, they will happily answer. Yeah, please.
Andrés, I got a little lost in those hypotheses of predation, hybridation, and the supplementary homeostasis. I didn't get so much why hybridation was expected to change homeostasis.
Yeah, so the idea is that if they have no reason to develop mechanisms that control their body chemistry, because they already find the body chemistry they need in their food, there's no need for stoichiometrics. So, for example, if you do the analogy with temperature, the reason why lizards are not homeostatic is because they can find a place with the right temperature. They just go to the place with the right temperature. Right? They go in the shade, they go in the sun. You know, so that's the idea. You could argue it many ways, but this is one of the arguments.
It's really one of the constraints for the hibernation guppy is the amount of time it's got to forage. Because it's got to avoid the predation. So it will be intrinsically looking for high quality food because it had so long to forage.
Yes, and I think the generalization of low predation should be more homeostatic and hibernation should be less homeostatic was a bit naive. Now when we sort of start to propose it. And the result that we got points us at the subtlety that both of them actually follow the same. Both of them are homeostatic, but with the nutrient they have limited. So they are so hibernation actually are very homeostatic here with respect to nitrogen. So what you're saying could very much be right. They both are limited by food, just by different nutrients. Yeah.
Another question. You said at the start, I thought you were promising us that you were going to get into what it means for the environment.
Yeah.
All you told us about is what it means for the fish.
Yes, and sorry for the false promise. No, this is the part of what is important for the fish is the part that's coming that we haven't started doing yet. But there's two avenues here that we're going to put. Because first we wanted to see are these traits variable, or they're not variable, and can they evolve? If they cannot evolve to an evolutionary biology, there's no equilibrium feedback, so the consistent function is not that attractive. But one of the things is do selection lines now on these traits and select for those traits and then put them in a mesocosm environment and see how nutrient recycling differs between the two environments. Second, in the wild, what we actually have been doing, but it's a long-term process, in those long-term experiments, we're doing isolate tracer addition experiments, where we're going to just reconstruct nutrient cycling before the guppies went in, after they went in, and after they evolved. So that's sort of like, it doesn't answer the question, it just answers what tools are we going to use to answer that question, but that's where we are.
And then, what are you going to do about the tricky question of the teleology of this? Are they doing it on purpose? Are they changing things on purpose? And that's the thing that we're really interested in our work.
I am very much of an atheist when it comes to that, in the sense that there's no teleology in evolution. There's what happens. It happens because of the selective pressures and the genetics of it. I don't think there's any conscious effort of the guppies to excrete more or less because that way they will have a better or worse environment. At least that's the assumption in the whole program, is that that's not happening. I know that some people think more in people think more of like terms like niche construction, things like that. They do think a little bit about teleology more. I guess the short answer is I'm not from that school of thought. As an evolutionary biologist that I am, I'm very much like mechanistic, so to speak. Yeah.
But then you get into situations in which, well, our situation is these the shrimps that are working on the periphery. And what we find is that the shrimps work really hard on the part of the periphyton that they don't actually ingest, that they don't assimilate. They ingest it. They regard and all that stuff. So why are they doing all this work in cleaning the environment? You've got the shrimps.
Yeah.
They do all this cleaning. Now, does it mean that they are trying to produce a nice larva of algae? And that's a possibility, isn't it?
No, it is a possibility, and there could be, there's such thing as prudent predators and, stuff like that. It's just, I don't think you need to necessarily invoke a high level of consciousness for that to happen. It's just, if what happens has a benefit today, it will be selected by natural selection. But yeah, it's fine. No, I know about the shrimps. It's fascinating. Yeah.
Another question.
Yeah.
You are looking for some difference in roughly period, both are in roughly period. But once we have two kinds of ecotypes, I think it's also expected a kind of behavioral homeostasis inside them.
Mm-hmm.
So I don't get. I think I don't get. Why did you expect this rapid change over time?
Why did they expect this rapid change? Well, no, because what we were doing is putting high predation guppies into a low predation site. We were perturbing their environment. And that's when we would expect a rapid response.
Okay.
Yeah.
So it's not evolutionary change over time.
No, if it's stable, it shouldn't evolve rapidly,
Okay.
obviously. I mean, of course, things are not stable these days, so rapid evolution is more relevant than ever. But, yeah.
Okay.
So first, I would like to thank you for your talk.
Thank
You
you.
can see that you are very passionate about it, which was a beautiful talk.
Thank you.
And I don't have like proper questions, but I have some curiosity. The first one
It's the best.
is the guppy's colors change in the high predation and low predation side? But do you know what could explain this? If
Predators, the visual predators.
Visual predator.
So they're drabber, they're less colorful in high predation sites, and they're more colorful in low predation
Oh,
sites.
nice.
So females have a strong preference for higher color, but so do predators. So, so that's the, yeah.
Yeah. What's your best experiment? Did you run it in their native range?
Which one?
The
The introduction?
the base.
The backy,
Yeah,
yes. Now we wouldn't go introduce them somewhere else now.
backy.
In fact, we have decided we're not doing more, even within. So.
And I was curious about does their homeostasis may change depending on their native or no native range? Like, if that happens?
I have no idea because we never looked at it. But
So are you
well, one interesting thing about a different angle that we're looking at, or essentially that Jefferson is leading, is looking at invasiveness and urbanization, and this whole hypothesis that urbanized environments are highly nutrient-rich environments, and there's a lot of questions about how would these hypotheses play in those environments, and what do they do with that excessive nutrients, etc., etc. So, yeah, that's a really interesting question, and one that we're only just starting to think about, really.
I have one question and one that's kind of extrapolating.
Okay.
The first one is, in a low predation environment, the multicolors tend to be brighter the adaptation to predation, and it's a sexual adaptation because the seniors
Mhm.
it could be linked to the captive guppies we have because like they're all over the place. So this could be the mechanism that created the guppies that we have in captivity.
They have huge diversity of color patterns, and it's due to many different loci. And people have studied that since the 40s, 50s, like the genetics of coloration of guppies, and this from a Mendelian perspective. Um, so yes, they were interested precisely because they were interested in creating fancy guppies. And yeah, that genetic diversity of coloration is what leads them to be a great species for domesticating and creating all those colorful patterns that you see in the pet shop. Yeah.
So the captive universe kind of mimics a more predation site.
Maybe it's. I mean, it's artificial selection. So I mean, they just like pick the guppies that are more colorful and they breed those, right? So it's not exactly natural.
Like total outside guppies, but
Okay.
like animal killifish, really.
Yeah.
Their environment's like super nutritious, like a lot of microorganisms, a lot of manifestations, archaea, exotic, stuff like this. And they have a low predation because the water is like very muddy and so just like the arthropod predation that they would have some heavy nutrient recycling in the first place they did
Mhm.
because they eat all the microorganisms in the puddle
Mhm.
and later they die. It's almost like I don't know salmon like when they have the
Yeah, I know about those fish. Yeah.
so maybe in the dry season they could contribute with the nutrients in the floodplain.
Yeah. Sure. I mean, especially good. Yeah, I don't
Some
know.
study about it.
Oh, I don't know. I don't know. I know how people have looked at the input of nutrients like salmon from their death, and it's a significant contribution. I don't think anybody has looked at the contribution of like killifish animal death to recycling.
The killifish haven't brought the nutrients in. So the difference with the salmon is that they brought the
Yes.
nutrients from
Salmon is
the
not really
ocean.
recycling. Yeah.
So the comparison with killifish is interesting, isn't it? Because like the killifish, the pool of nutrients is there and would have done, isn't it? But whether they've made them more
Whereas that
available,
available to the land.
Yeah. That's interesting. That's true. The salmon is not a great analogy there. Yeah. All right. Well, um, I know I'm short, but I didn't want to make a long talk because I wanted to just open up the discussion and I wanted to not bore you for too long. But thank you so much. You've been so welcoming so far, and yeah, I'm having a great time here. So thank you for coming.